Activity 1. To make sprouts. Sprouted dal is an extremely healthy food. To make sprouts, soak some moong or gram seeds in water overnight. Drain the excess water and tie these seeds in a moist cloth. Keep this cloth moist. In a day or two, you will see small white shoots growing from it. These are called sprouts. You can make a very tasty salad by adding some chopped onions, tomatoes, coriander, lemon juice, salt, and powder of pepper and cumin to it. Food chain. All living organisms depend on each other for food, either directly or indirectly. Plants are called producers, as only they produce food, which is made available to other living organisms through the food chain. A food chain shows who eats whom. A deer, herbivore, eats grass, producer. The deer is eaten by a lion, carnivore, and when the lion dies, its body is eaten by vultures, scavengers. In this way, a food chain is formed. Activity 1. To test the presence of starch in food. Take a small piece of potato and put 2 to 3 drops of diluted iodine solution on it. The piece of potato will become blue-black in color, which shows that it contains starch. Potatoes have high starch content. You can also try this iodine test with some other food items and observe the results. Hand picking. The method of hand picking can be used to separate solids which are easily visible to the naked eye, that is, stones and pulses or rice, etc. Threshing. A paddy plant has a number of stalks on its upper portion. Separation of these grains is carried out by spreading and beating stalks on the ground. This process of separating grains from stalks is called threshing. Winnowing. Winnowing is a method of separating heavier and lighter components in a mixture with the help of wind or blowing air. The mixture of grains and husk is made to fall from a height. Grains, the heavier of the two, fall to a side, forming a large heap of their own. Husk, being lighter, blows away easily and form a separate heap at a distance. Activity 4. To know the solubility of sugar, salt and glucose. Take 100 ml of water in 3 beakers and label them A, B and C. Add a spoonful powdered sugar to the beaker A, common salt to the beaker B and glucose to the beaker C. Stir the contents of each beaker with a glass rod. When the common salt in beaker B dissolves, add 1 more spoon of salt to it and continue doing so till no more salt dissolves. Repeat the same process with glucose and powdered sugar. Record the number of spoons of common salt, glucose and sugar added respectively in each beaker till they could not be dissolved anymore. The difference in the number of spoons shows that different substances have different solubility in the same amount of water. Activity 5. To find out the role of temperature in solubility. Take a beaker, fill it with 200 ml of water, add 2 spoonfuls of sugar and stir it well. Continue doing so till no more sugar dissolves. Now start heating the beaker carefully. Stir the solution. After a few minutes you will notice that the sugar collected at the bottom of the beaker earlier has dissolved. This activity shows that solubility increases with the increase in temperature. Activity 7. To understand sedimentation and decantation, take a mixture of sand and water in a beaker. Allow the mixture to stand for 2 minutes without disturbing it. Sand settles at the bottom, sedimentation. Now slowly and carefully transfer clear water to a beaker, decantation. Activity 6. To separate insoluble solid from liquid using filtration method, in a 100 ml beaker, take some tap water and add a spoonful of soil dust into it. You can also make a mixture of sawdust and water. Pour this water and mud mixture through a funnel made from filter paper. Observe that mud settles down on the filter paper while water, muddy but without any solid particles, collects in the second beaker. Loading. The process of separating suspended impurities by making them heavy is called loading. At times we get muddy water in our taps. To clean water in a short time, a crystalline piece of alum is moved along the surface of the muddy water. Some alum gets dissolved in water. Its particles get attached to dust particles and make them heavy. These heavy particles make them settle down faster, resulting in earlier sedimentation. Distillation. Distillation is a process of separating two miscible liquids by using the methods of evaporation and condensation simultaneously. If two liquids in a mixture are miscible and there is a difference of 15 degrees centigrade to 20 degrees centigrade in their boiling points, they can be separated by distillation. We set up the distillation apparatus called Liebig's condenser with the distillation flask containing mixture. When the flask is heated, the liquid with the lower boiling point vaporizes first, gets condensed and collected in the beaker placed under the outlet of the condenser. The other liquid remains behind in the distillation flask. This method of distillation can also be used to separate a soluble solid from a liquid solution. Separating funnel. Separating funnel is round and broad at the top and narrow at the bottom. The lower end has a stopper to control the flow of liquids inside the funnel. Mixture is poured into separating funnel from top. When immiscible liquids are poured, two layers are formed with the lighter constituent remaining on top. Using stopper, liquid in the lower layer, which is heavier liquid, can be collected in a beaker. The flow is stopped as soon as the lighter liquid in the upper layer reaches the stopper. Activity 1. To know what is a thread made up of, take a sample of any fabric and pull a loose thread, yarn, from one of the edges gently with the help of pin or needle. You'll find that fabric is made of yarn woven in a crisscross fashion. Press one end of yarn with your thumb and scratch the other end of the yarn along its length with your nail or pin. The yarn splits up into thin strands. The strands of yarn or thread are made up of still thinner strands called fibers. Activity 3. To separate soluble and insoluble substances. Take 6 test tubes with a level spoon of sugar, face powder, sand, chalk powder, sawdust and washing powder. Add 50 ml water in each test tube. Shake vigorously and keep aside undisturbed. See which substances are soluble and which are insoluble. Record the observations in your notebook. Activity 4. To know the materials which are heavier or lighter than water. Fill half a beaker with water and place a wax candle, a pencil, dry leaves, green leaves, an iron nail, a plastic toy and some stapler pins in it one by one. Find out which of them float or sink in water and classify them accordingly. Activity 1. To check whether a given change is reversible or irreversible. Take a spoonful of sugar and dissolve it in half cup of water to get a clear solution. Pour it in a pan. Now evaporate water over a burner. You will recover the sugar settle at the bottom of the pan. What kind of a change is it? It is a reversible change because we have got the sugar back on evaporating water. Activity 2. To observe a chemical change, burn a piece of paper. You will notice that a lot of smoke is produced and ash is formed. Ash has different properties than paper. Hence, this is a chemical change. Activity 3. To understand the effect of cooling of air. Inflate a balloon and measure its size with a measuring tape. Keep it in refrigerator or in a bowl of ice cold water for some time. What do you observe after some time? The balloon will shrink, that is, it will contract. Actually, the air filled in the balloon contracts on cooling, thus decreases its size. Photosynthesis. Green plants have chlorophyll, with the help of which they can make their own food from carbon dioxide and water in the presence of sunlight by a process called photosynthesis. Therefore, plants are called autotrophs. Living things show movement. The movement of an organism from one place to another is called locomotion, and the change in the position of any body part is called movement. Animals move around in search of food and shelter. There are various locomotory organs like legs for walking and running, wings for flying, and fins for swimming. Thus we can say that animals show both movement of body parts and locomotion. Plants remain in a fixed position, but they show movement of their parts. The plant movements are called tropic movements or tropism. For example, a sunflower always faces the sun. Activity 1. To prove that the exhaled air contains carbon dioxide, add a few drops of lime calcium oxide to a test tube containing water. Shake it thoroughly to get a clear solution. This is called lime water. Cover the mouth of the test tube and gently blow into it through a small gap. What do you observe? You'll observe that the lime water in the test tube turns milky. It proves that exhaled air contains carbon dioxide. Activity 1. To know how roots anchor the plants, take a bowl and place some cotton wool in it. Put some seeds of gram in it and sprinkle water. Continue to sprinkle water for a week till small plantlets appear. Try to pull out the plantlets carefully. You will see that the roots are clung to the cotton wool and it is difficult to pull them out. Parts of stem. Node. It is a point on the stem from which a leaf arises. Internode. The portion of the stem between two consecutive nodes is called an internode. Axil. The angle between the stem and the branch is called an axil. Bud. It is the part of a young stem protected by overlapping young leaves. Activity 2. To show movement of water through stem. Take 2 buckets, A and B, of fresh similar flowers with long stem. Crush the tips of flower stems in bouquet B. Turn up the tips and tie them up tightly with a sellotape. Dip the flower stems of bouquet A and B in separate flower vases containing same amount of water. Observe the setups for 4 to 5 days. Do you observe any difference between the flowers of bouquet A and B after 4-5 days? Is there any other change in the flower vases in which the flowers were kept? Activity 3. To show how water moves through stem, cut one end of the stem of a balsam plant or any other herb with tender and translucent stem with a sharp blade. Dip the end in a glass or beaker containing colored water. The water can be made colored by adding a few drops of red ink or saffron into it. Observe its stem, leaves and flowers the next day. Cut a slice of the stem with a blade and observe it under a hand lens. Where do you see red color in the slice? You will see red coloration in the cross section of the stem. Parts of leaf. Petiole. The slender stem or stalk which supports the blade of a leaf is called petiole. Leaf apex. It is the tip of the leaf. Leaf margin. It is the outline of the leaf. Lamina. The main body of a leaf is called lamina. Veins. Veins are narrow vessels which carry water and nutrients to the leaves. Midrib. It is the thick vein in the middle which runs from the base of the leaf to its tip. Activity 5. To test the presence of starch in a leaf, put a leaf in a tube. Pour enough spirit into the test tube to cover the leaf completely. Place this test tube in a beaker or water bath half filled with water. Heat the beaker. Keep heating till all the green color of the leaf comes out in the spirit. Take this discolored leaf out and wash it with water carefully and perform the iodine test. The leaf turns blue-black due to the presence of starch. Activity 6. To show the need of sunlight for photosynthesis, keep a potted plant with green leaves in the dark for a day or two. Cut a strip of black paper. Cover a part of a leaf completely with this strip of black paper so that it cannot receive sunlight. Now leave the plant out in the sun for a few days. Take the leaf with the black strip, remove the strip and perform the iodine starch test. The part of the leaf which was covered with black paper does not turn blue-black when treated with iodine. This proves that the starch is manufactured in the leaves by a process which needs sunlight. The part of the leaf covered with black paper could not manufacture starch because it was not exposed to sunlight. Activity 7. To show the process of transpiration, take a healthy and well-watered herb. Place it inside a polythene bag and tie up the mouth of the bag tightly. Take another pot and fix one wooden stick in the soil. Tie a polythene bag also around it. Keep both the pots out in the sun for about 3 to 4 hours. Open the bags and see inside. The bag with the herb placed in it has tiny droplets of water clinging to the inner surface of the bag. No water is seen in the polythene bag containing wooden stick. The herb tied up in the bag loses water vapor from its pores, stomata. This vapor condenses on the inner surface of the bag to form tiny droplets of water. Activity 8. To observe inner parts of ovaries, take ovaries of any 2 flowers. With a blade, make a horizontal cut in one and a vertical cut in the other. Put a drop of water on each to prevent them from drying. Now, observe the inner parts of the ovaries with the help of a magnifying glass or a microscope. Write your observation in your notebook. The skeletal system. The framework of bones inside our body forms the skeletal system. It protects internal organs and helps in the movement of different parts of the body. The skeleton of an adult human has 206 bones. Functions of the skeletal system. It gives shape and support to the body. It provides a hard framework to protect soft organs and muscles. For example, the brain is protected by the skull. The heart and lungs are protected by the rib cage. The skeletal system, along with muscles, helps our body parts to move. Bones contain bone marrow, a soft substance inside which blood cells for the body are formed. Parts of the skeletal system. The human skeleton can be divided into 4 parts: the skull, the backbone, the rib cage, and the limbs. Activity 2. To measure the length of bed, you can measure the length of your bed using a non-stretchable thread or ribbon. Take a thread or ribbon and put a mark on it. This length would be the standard of measurement of length called the string length, L. Now using the thread, you can measure the length of the bed. Length of the bed in this example is 1.5 string length, that is 1.5 meter. Activity 3. To measure the length of a curved line, draw a curved line on a chart paper. Take a thread and tie a knot at its one end. Put it at one end of the line. Place the thread along the curved line, stretching and keeping it tight till you reach the other end. Mark the thread on this point with ink. Now you can measure the distance between 2 points with a ruler. This will be the approximate length of the curved line. Activity 1. To show how chemical reaction produces electricity, take 2 small pieces of wire and peel off their ends. Tightly coil one end of a piece of wire around a zinc strip. In the same way, wind one end of the other wire around a copper nail. Stick zinc strip and copper nail into a lemon. You have prepared a simple electric cell. You can test the working of this arrangement. Connect a bulb between 2 free ends of wire and darken the room. You'll find a feeble glow being emitted by the bulb. Electric current is generated due to chemical reaction of copper and zinc with acid in the lemon. Lemon juice acts as an electrolyte and is capable of generating electricity. Electric bulb. An electric bulb is made of a very thin and fine quality glass which is fixed on a metal base. There is a very thin wire inside the bulb. This wire, which is made of tungsten, is called filament of the bulb. On passing electricity, this filament heats up and glows to give light. The properties of tungsten ensure that when filament gets white hot, it does not melt. This tungsten wire is twisted to form a tight coil. As this wire is thin, long and heavy, it is supported on 2 thick and strong wires called supporting wires. One of these thick wires is connected to the metal tip at the center of the base. This forms the 2 terminals of the bulb. These terminals do not touch each other as the space between them is filled with sealing wax. Activity 2. To know about proper connection, connect a cell with a bulb using connecting wires as shown in the figures, one at a time. Does the bulb glow in any of the cases? No. Now connect the bulb as shown in the diagram on the right. Does the bulb glow now? Yes. The bulb will only glow when 2 terminals of the cell are properly connected to the bulb. Activity 3. To make an electric circuit, consider different circuit connections as shown here. The bulb glows in figures 4 and 7. Why? This is because the electric circuit is complete in these 2 connections only. In all other connections, circuit is not complete and hence the bulb does not glow. Electric switch. There is a switch in a circuit to open, break or close the circuit. When a switch is on, the passage of electricity starts, and when the switch is off, the passage of electricity stops. Observe the picture of a switch in the given picture. When you push it down or into on position, the two pieces of metal touch, closing the circuit, and the bulb glows. When you leave it, the springy metal bounces up, the circuit is open, and the bulb turns off. Activity 4. To know how a switch works, make an electric circuit as shown in the diagram. Fix a metallic sheet with cuts at both ends between the fixed pins. The bulb glows. Now remove the sheet from one of the pins and see what happens. The bulb does not glow. The metal sheet acts as a switch, covering the gap between the pins and completing the electric circuit. Electric torch. A torch is a device in which simple circuit is used to get light. An outer plastic case to provide insulation. Two cells with positive terminal of one connected to the negative terminal of other. These two cells, when connected correctly, complete the circuit and provide a path for electric current to flow. A slider to switch the torch on or off. When the slider is in on position, a small strip of metal moves and connects with another metal connector. Metal spring to make contact with the switch and complete the electric circuit. This is attached to the negative terminal of the lower cell and connects it to the base terminal of the torch bulb. A reflector is placed around the bulb so that the light from the torch covers a great distance and is also much brighter. A torch bulb works just like an electric bulb. It also contains a filament that heats up when the circuit is closed to provide light. Activity 5. To test whether an object is a conductor or an insulator, make an electric circuit as shown. Now place different objects like glass, rubber, plastic, copper wire, iron nail, brass screw, pencil, wood, etc. between the two crocodile clips. Does bulb glow in all the arrangements? Write down your observations. You will find that metals, not all, are good conductors and rest are insulators. Activity 1. To prove that magnetic effects also act in different mediums, place a paper clip on a page of your notebook. Place a magnet just below the clip from the other side of the page. Now move the magnet up, down or left-right. You will see pin also moves on the surface of the page. You can do this activity using plastic or wooden sheets. Activity 2. Activity 3. To test attraction and repulsion in magnets, suspend a thin bar magnet as shown. First, bring the north pole and then the south pole of another bar magnet near the north pole of the suspended magnet. See figure 1 and 2. What do you observe? Now suspend a thin piece of iron as shown. Repeat the above steps by a bar magnet. See figure 3 and 4. What do you observe? We find that two magnets can attract and repel each other depending on similar or dissimilar poles. However, iron is attracted towards both the poles. Activity 4. To prove that a magnet always has two poles, cut a thin strip from a magnet. Introduce one of the poles, say north, of another bar magnet near any one end of the thin strip. Attraction takes place. Now introduce the same pole, north of the bar magnet, near the other end of the thin strip. Repulsion takes place. Divide the thin strip into two parts and repeat these steps. Observations in each case will remain the same. Activity 5. To prove that a magnet always takes north-south direction, Suspend a thin bar magnet horizontally so that it can rotate freely. Wait till magnet comes to rest. It will arrange itself in a north-south direction. Rotate the suspended magnet and you will find that it again comes to rest in north-south direction. A freely suspended magnet always points in north-south direction due to its directive property. Activity 1. To examine the rectilinear propagation of light, keep a lighted candle on a table at one end of a room. Look at the candle through a straight pipe. Is it visible? Yes, it is. Now bend the pipe a little and try looking the candle through the pipe. Is the candle visible now? No, it isn't. This shows that light travels in a straight line. Activity 2. To verify the rectilinear propagation of light, take 3 cards with fine holes at the same height in the center. Use modeling clay to hold the cards in upright position on a table. Line up the cards so that all the holes are in the same straight line. Place a lighted candle on the table whose flame is visible through the holes on the other side. Now shift 1 card from its position. Can you still see the candle flame? No, you cannot. The light of the candle is visible only when the cards are in a straight line. This proves the rectilinear propagation of light. Activity 5. To prove that light changes its path after reflection, ask one of your friends to hold a mirror in his or her hands in a dark room. Direct a beam of torchlight on the mirror. Now, adjust the direction of the torch so the patch of light falls on another friend standing in the room. The activity suggests that light travels in a straight line and changes direction after getting reflected from a mirror. Activity 6. To prove that a mirror gives laterally inverted image, write your name on a sheet of paper in capital letters and hold it in front of a mirror. You will find that some of the letters reflected in the image seem correct while others seem wrong. This is because of lateral inversion. You may have seen an ambulance. The letters in front read ambulance. This is done so that the driver of any vehicle in front of the ambulance is able to read the word ambulance correctly in his rear view mirror. Activity 1. To understand condensation. Boil water in a saucepan. You will see water vapor is formed. When the vapor comes in contact with the cold surface of the plate, it condenses to form tiny droplets of water. Thus, when water vapor comes in contact with a cold medium, it gets condensed. Activity 1. To prove that air occupies space, insert an inverted glass tumbler in a bucket of water. Do not tilt the tumbler. Does water go inside it? No. Now tilt the glass. What do you see? The water rushes into the tumbler and bubbles are formed in water. These bubbles are formed due to air rushing out of the tumbler. In the first case, water could not enter because of the presence of air inside the tumbler. This shows that air occupies space. Activity 2. To prove that air has mass, take a measuring scale and tie a string at its midpoint. Tie 2 balloons at an equal distance from the midpoint so that the scale is balanced. Now fill air in one balloon and place it at its original position. The scale tilts towards the side of the inflated balloon. Why? The inflated balloon has air inside it, and this makes it heavier. This shows that air has mass. Activity 3. To show that air contains water vapor and dust particles, take a glass tumbler and fill it with ice cubes. Water vapor present in the air condenses to form droplets around the cold surface of the tumbler. It proves that air contains water vapor. Close the door and windows of a room and make it dark. Allow a beam of light to enter the room from a small window. You can see dust particles. It proves that air contains dust particles. Activity 4. To prove that air supports burning, light a candle and put a glass tumbler upside down over it. What happens after some time? The candle extinguishes. Why? The amount of air present in the glass tumbler is used for burning, and when air or oxygen is exhausted, the candle extinguishes. This shows that air supports burning. Oxygen cycle in nature. Human beings and most of the animals take in oxygen and give out carbon dioxide. Plants also take in oxygen and give out carbon dioxide during respiration. But plants carry on the process of photosynthesis during the day. In this process, plants take in carbon dioxide and give out oxygen. In this way, oxygen is recycled in nature by plants. The main reason behind the increasing quantity of carbon dioxide in the atmosphere is cutting down of trees indiscriminately. If it is not checked, there would be a great imbalance in the quantity of oxygen and carbon dioxide in the atmosphere. Nitrogen cycle. Nitrogen is required by plants and animals for growth. However, neither plants nor animals can absorb nitrogen directly from air. Nitrogen from air gets fixed in the soil in usable forms as compounds with the help of certain nitrogen bacteria. These bacteria are present in roots of leguminous plants like peas, beans, peanuts and soya. The compounds of nitrogen are then absorbed by the plants from soil. When animals eat plant products, they get nitrogen from them. Activity 5. To show that water contains oxygen, fill half a beaker with fresh water and heat it. After a few minutes, observe the base of the beaker. Tiny bubbles are seen at the base. Oxygen gas, which is a component of air, is dissolved in water. On heating, the solubility of a gas decreases. Hence, oxygen is released in the form of bubbles. This shows that oxygen is dissolved in water. Activity 6. To show that soil contains oxygen, take a beaker half filled with water. Pour half cup of soil in it. After a few minutes, you will see that bubbles of air start rising from the surface of the soil. This shows that soil contains oxygen. Activity 1. To show that biodegradable wastes decompose easily. Separate all food waste, vegetable and fruit peels, eggshells, tea leaves, etc. and waste paper from the garbage collected at your home. Take an earthen pot and fill it with this waste. Cover it with soil. Dig out the garbage after a week and check if the garbage placed in the pot has rotted completely or not. You will find that the garbage has decomposed completely. It shows that biodegradable waste decomposes easily. Composting. When biodegradable waste decomposes, it gets converted into simple substances or manure or compost. A layer of biodegradable waste is buried in a deep pit. The waste is then covered with a layer of moist soil and watered. The process of layering is repeated until the pit is completely filled. The pit is left undisturbed for a couple of months. The microorganisms present in the soil feed on the organic waste and convert it into compost. Compost is rich in plant nutrients and is therefore used as manure. Unlike chemical fertilizers, it does not damage the quality of soil over a period of time.